All right, so I'm just going to touch on slide number 3, and then but we're jumping to slide number 4. So we have cancers, and we had said that we're talking about these different controls on the cell cycle, and some of them are there to speed up the cell cycle, and some of them are there to slow it down. And so we have some braking mechanisms, which, it's going too fast, we don't need so many cells, or we need more cells, speed it up, buddy. That's acceleration mechanisms. And so remember, we said that in the skin, you have stem cells that will undergo cell division and produce a new skin cell, and then you still have a stem cell to be stimulated again at a later date. And these things get sped up and slowed down. Okay, so we have, I always have to look at my slide here for just a minute, and it ends up that there's a Dr. Epstein who has done a lot of the work on basal cell carcinomas and this condition that Patricia has. So normally we had said with basal cell carcinomas, typically it's from overexposure to sun. And it's like a delayed thing, as in 10 years. It's not a rapid thing. It's delayed by some years. And here we are with this young lady who was 17 years old, and she was already having problems with skin cancers. And so it ends up that there is this gene called the patched gene. And we have a mutation in this gene. And so the patched gene is a gene that provides protection against skin cancer. And the product of this patch gene is a receptor for a growth factor. And the funny thing is that this growth factor that will interact with the patched gene receptor is something called sonic hedgehog. So growth factors, cell types, and things, they're named by scientists. And whoever named this growth factor had a sense of humor. So it speeds things up. So he said, we'll call it sonic hedgehog. So sonic hedgehog is the growth factor that combines with the patched gene. And this Dr. Epstein found that in Patricia, she had a messed up patched gene, a mutated patched gene. And so we had said we have this sonic hedgehog that normally would tell the cells to start dividing. Well, it ends up with Patricia, even when sonic hedgehog wasn't present, her receptor thought it was. And so it was constantly saying, we need more skin cells. And that's the problem that she had, is that her patched gene was defective, and it would think that sonic hedgehog growth factor was there when it wasn't. And so she would produce more and more of these cells, and they would pile up, and we'd have a tumor. And she'd have to have it cut off. Okay, let's see where we are now. All right, so we've already mentioned some of this stuff. We had said we have both internal and external signals that are affecting the growth, the cell cycle. And we had said we have internal signals. We had said, during the S phase, the S phase, as the DNA is being replicated, you end up building up a chemical called a cyclins. And these cyclins, you build up as the DNA is getting replicated. You've got, instead of 46 chromosomes, you now have 92, because you have two sets of your chromosomes, because one has to go to each of the two daughter cells, right? So because of this, as it's doing this S-cyclin builds up, and so it tells the cell, hey, it's time to go to G2. We're done with this replication process. And then we have the M cyclins. And so what did we say during G2 is going on? We're making spindle fibers. And so you build up this other kind of cyclin, the M cyclins, and say, yes, we can go to mitosis. And so we have these chemicals, these internal chemicals that are saying, time to go, time to move ahead to the next stage in the cell cycle. And we also have these growth factors like Sonic Hedgehog saying, we need to speed up cell division. So those are external controls, Sonic Hedgehog, and the internal controls are those cyclins, and there's something else called a kinase. Cyclins and kinases are the internal stuff that says, hey, we can go through mitosis. Yeah. Okay, so we have these checkpoints. So when we think of that cell cycle, we have checkpoints. We have this G1 checkpoint. This is an internal signal. It says, okay, do we have enough of these cyclins and kinases built up? Time to go into the next stage. The G2 checkpoint, making sure that we've gotten all that DNA replicated. And then at the end of mitosis, we have the mitotic checkpoint. So let's see. So we have the G1 checkpoint. Remember, okay, so I've flipped to slide number 8. So remember, we said the G1 checkpoint has to do with going on and going through cell division. What happened? At the end of G1, we had two options, right? You could keep going or you could leave G0. And we had said, what is G0? That's a cell that will never go through cell division again. But if you go through the G1 checkpoint, what happens? You start replicating the DNA in the synthesis. So you check, okay, at the G1 checkpoints, you check the integrity of the DNA, and if everything is good, you keep on going. You keep on trucking, yeah? And then at the G2 checkpoint, you make sure you replicated all of that DNA and that it isn't damaged. And if it is, you got to fix it first. And then the mitotic checkpoint. We said in mitosis, you got to make sure that you've done everything right. So between metaphase and anaphase, you got to be sure that all of those chromosomes are connected to the spindle fibers. And that is what the mitotic checkpoint is. Okay, keep on rolling here. So we've got a nice little picture of all of that. Good. And then we have another picture on slide 10. And you notice we have some colored squares, colored blocks and balls, circles and squares. And what it is, these are those cyclins or kinases and cyclins, and they have to build up to a certain level. That's the sort of checking that's going on before you move to the next stage. All right, so we have these things like I've I kind of said, enough of that. We moved to slide 11, okay? So we have cell cycle signals. We say external signals, epidermal growth factors. Sonic Hedgehog is one of those. And, oh, we have some other stuff here. One of the things that non-cancerous cells, if you work with cells in cell culture in a lab, one of the things you know is that cells will only grow for a little while and they stop. Ugh, drives you crazy. And why is it? It's because they bump into each other. They have something called contact inhibition. And so they'll only divide just so many times in culture because they bump into each other. And then there's this other thing, the ends of the chromosomes. We've said the chromosomes are these giant molecules, right? And the ends of them have these repeating units. And so there are no genes out there, but there are these repeating units called telomeres. T-A-T-A-T-A, so a T, T-A, 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 a whole bunch of these at the ends of the chromosomes. These are called telomeres. Well, it ends up, every time you go through a cell division, you lose some of these. And these repeating sequences, as you lose these from the ends of the chromosome, the cell stops going through cell division. It'll only go through it about 70 times in cell culture. Okay, so one of the things, I didn't even mention it. I saw it on my slide and I didn't even mention it. At the end of that G1 checkpoint, I'd said that you check to make sure that the DNA, check the integrity of the DNA. Make sure it's not messed up. And what happens if it is messed up? Well, not only do you not continue on, the cell commits suicide, something called apoptosis. Apoptosis is a way, if a cell senses that it's corrupted, it kills itself. And so it's something called programmed cell death, and it can be unleashed by either internal or external signals. And in some cases, it's maybe a cell is becoming cancerous, it's got a corrupted DNA. In other cases, in development, let's talk about development. What about frogs? When a baby frog is born, it's not a frog, is it? It's a tadpole, and a tadpole has a tail. And when the frog metamorphoses from a tadpole to a frog, it loses its tail. Well, how does it lose its tail? Apoptosis. I just realized I've gone 13 minutes. I better stop right here. An apoptosis is when you and I were just a little wee fetus in your mama, we had limb buds. We didn't have fingers. We had clubs at the ends of our arms. Apoptosis. Produced, you say, look, Mom, I've got fingers. Okay, I'm going to stop right here because we've gone way too long.